All right, so as I said, today's session here is on recording assessments within ARM software primarily. So main topics we want to cover is data entry within the software, talking about describing the assessment that was done. Then we'll talk about a couple of tools for kind of data confirmation once the data is in ARM. What do we do to make sure that we entered it in correctly, and get just a little flavor of standard evaluations to kind of standardize the way that we describe assessments. So first thing first, we'll start off with really, I guess, how you would start with an assessment. And that is to, really how do we describe the assessments? So I'm just going to open up what amounts to basically a totally empty trial here. And I'm already on the assessment data section. So the editor here is really for filling in data. So when we go out to the field and take an assessment, each individual rating, action that you would perform across all of the plots in the trial gets saved in its own column in this assessment data editor. And so really this screen has two components. So really your assessment has a description of specifically what was done and some details about the site and the crop and pest, things like that at that time of assessment. And so we refer to all of that information as the assessment header. So we'll refer to this top portion of the editor really as the header describing the assessment that is done. Then down below, we actually have the data. So the spreadsheet, if you will, to put in the numbers that are being recorded for each plot. So to start with then, it's really a matter of, what do I fill in? How do we describe the assessment that we're performing? As I scrolled here, you can see there is a large number of rows. You might not see all of them to start with. Some of them can be hidden. So over on the right, on the properties panel, I've already showed all of mine. So I have the all fields selected previously, but there are some options, and you certainly usually won't see all of them. Let me just pick one here that will, I'll just grab one that'll hide some of them. So this is now quite a bit shorter. So if you don't see something we're referring to today, you'll find on your properties panel a hidden fields box in this corner, and that will show different rows that are available to use, but just at this moment are not visible. So it can be easy to specifically choose, oh, I need to see my rating date, and I'm looking for my crop height. Oh, this is where I can fill in crop height, etc. Or down below here in this views section, you can do something like all fields. So you can see every single row that's possible to record. And that's kind of what I'll use here, just so we can kind of see the full gamut of things we can utilize. So just, I guess, trying to talk through how to fill in an assessment without having an example is pretty hard. So I'm just going to take a really basic a pest stand count. Maybe it's the same day as an application that we're going to perform. Trying to be as general as possible. Obviously, with each different kind of research, there's a whole host of possibilities here, but we'll just stick with this really basic one just to talk through maybe the thought process behind how to fill it in and what to fill in this header section. So the first few rows really are the pest and the crop. And so typically I tell people in most cases, you probably only need to fill in one or the other. It would really be which object are you planning to rate? In our case, if we're wanting to have a stand count of how many weeds are out in in the plots before we make that application to get rid of them all, hopefully, then the pest would be the focus of that particular assessment. Because of course, within the trial itself, we are going to have an entire section devoted on describing the crop and a section devoted on filling in all the different pests that we have present. So you don't have to necessarily describe the crop every single time if we're not even rating the crop at all. So maybe in our case, that's what we'll do is focus in on just the pest. In my case, I guess I said we're counting weeds, so I'll just take a weed here from the list. I've got pigeon grass for some reason in my favorites, so we'll just use that as the pest that we're wanting to count. But conversely, we could have filled in the crop if if our main focus of the assessment was the crop. Moving down, we have the rating date. So once we are actually taking that assessment, this is a very important field to put in. So if today we are taking that date, I press the space bar, that gives you today's date as a little shortcut. Otherwise, the drop down button here will give you a calendar to choose from. And as we move down, we'll come back to the SE name. That's a great field for when we're utilizing standard evaluations. But I do want to point out the SE description and actually compare that to I guess I'll call it the regular description right up here. So for years, this description was visible by default, and I will admit, anytime I trained on it, I told people this is a great way to fill in your your general human readable description, I guess, for the assessment. Well, it turns out that's actually not what this field was originally for. This is actually tied to the crop. We had the crop name, variety, and then it's really supposed to be the crop description. So it has nothing to do with the general assessment. That is supposed to be the SE description, which I think is a little confusing for a name because, of course, right now I'm not planning on, I haven't created an SE, so what do I even need the SE description for? So I think in the future, we're looking to find ways to make this a little more intuitive. But in general, now kind of trying to spread the word that description isn't really what you're looking for. It's actually the SE description is the best place for a simple, human readable shorthand, free text spot to just describe what is it you did. And that works very well as a little scratch pad. And you'll see now as we proceed that we do want to be very specific with how we describe each assessment. And this gives us just a nice way to quickly look and see what it was we did without having to be too technical. So that's the SE description instead of using the description. Next is really what I would call the big three. So if you didn't do anything else to describe what was done, I would say you need to use these three fields: the part rated, rating type, and rating unit. This is really the heart and soul of a description for a rating that is done. And so the part rated here is specifically the object that you are manipulating, you are rating, you are assessing, what have you. And you can see in this list that they can be very specific. You can have something general like the term adult, presumably for perhaps a bug, an insect that we're looking at. But we even then have a lot of times more specific in case we're only looking at male bugs or only wanting to count dead bugs or what have you. So you'll find that there's a lot of options in here, and the goal for that is to be very specific for the many different types of research that are being done. And it's maybe very important to distinguish what you're counting or what you are rating or looking at. And so in my case, it's very, very boring because we are literally just looking for maybe a plant. We're just looking at the plant in its entirety, doing the stand count. So I'm going to pick a pretty boring one. You could pick plant total, I suppose, if you were thinking we're really going to total up all the plants. I could also pick plant. There really isn't a wrong answer in most cases. I would say that the key here, don't get too hung up on making the right choice, but make the consistent choice. So if you're going to choose plant for your stand counts, then you'll want to always choose that one for all of the stand counts that you do. So that's really the trick, mostly when you're looking at that very lengthy list and trying to figure out how the heck do I choose one of these. And this part rated actually kind of has two components. So we picked the part. The second one, really there's only two options, either C for crop or P for pest. So just, are we referring to this part on the crop, or are we referring to this part on the plant? Or excuse me, the pest. So in our case, we were looking at the pests. So I'm going to choose P. Next one is rating type. So this is really, what did you do? I like to think of it as the action that's being performed. And again, as you can imagine, this list is lengthy and very very wide ranging in what is in there. But it's really, what is the assessment? What is it you're doing? And here, actually, I've already found kind of the area we're interested in, since we are simply counting. And then, of course, we do have, again, some options here. We have counting ones that have emerged, or many sorts of other different things that we could be counting. So you could choose to be very specific. And maybe this one just sounds like exactly what we're looking for, is how many weeds have emerged. Maybe that's more of a crop thing, or maybe, so I'll stick with something general. It's, again, kind of up to you and your group, but you want to do it consistently is the number one thing. You pick a value that, of course, makes sense for what you're doing, and choose that consistently. Then rating unit. I guess this is a great time to show that there's actually a lot of these lists. We have kind of two lists you may notice here right now. I have the full list that we've been kind of scrolling through, but then you can also create a favorites list. So this is a nice way to, instead of always scrolling through the massively long list of all possible options, you may find yourself only using, a handful of options based on your research. So a lot easier when you find one that you want to use, to simply add it to your favorites list. So then when you bring the list up next time, it will show you only ones that you have put into your favorites. And of course, you always have that option to display all of the items so you can find one that you haven't used before. So I'm going to filter on rating unit. I want to find number. And I guess I actually probably should have done it in the description. That's kind of that's the best place if you're trying to just search the list for something. You can either do a right click to filter for, or the box at the bottom here will also be a filter if you start to type. So that's a nice way to find something in these long lists. And so number is the unit I want, since count doesn't really have a unit like kilograms or meters or percent. It's just a number. So that is what I'm going to choose for my rating unit. There's also a minimum and a maximum. So sometimes that is auto-filled for you. If I bring up my rating unit list once more and just clear my filter, something like percent has a kind of a built-in minimum and maximum of 0 and 100. So some of these units you'll notice have that minimum and maximum that will kick in when we're actually filling in data. You can also hand fill it in if for whatever assessment you have a limit. In this case, it could be anything from 0 to, however many, so there really isn't a logical limit, but just for a demonstration's sake, we'll say 0 to 30. Just totally impractical, but we'll give it a try just to see how it affects our data entry in case you're using a 1 to 4 rating scale. Maybe another common example that you might have where there is just a practical limit of invalid numbers for your assessment. So that's the big three. That really can encompass the majority of describing what is performed. In some cases, though, especially with subsamples, you may need to use some of these, the sample size, collection basis, and reporting basis. Most, if you're doing simple subsampling, I just need to, pick 10 plants out of this plot to inspect or whatever. That sample size then is very simple. So something like that, I would just need to pick 10 and then plants or what have you. That sample size just describes what you're sampling. So you really have the objects and then of course the number of them. If you have a little more detailed subsampling, then you can utilize collection basis and or reporting basis. And so it's kind of hard to give some of those examples kind of get a little messy, I would say, trying to sort out exactly what you're doing. So I like to highlight the help, the user help. It's either the plus sign up here on the toolbar or the F5 key. And we'll see if it comes up. I have a separate monitor. Maybe this will maximize and it'll show up. Awesome. I have a third monitor for showing on Zoom, and sometimes it acts weird, but we got it. So this F5 help will pop up whatever box you have your cursor in. So I was clicked into this collection basis. The F5 help will show you a description of what ARM is expecting. What is this field for? And sometimes it'll have examples. In this case, it tries to give that definition, which is kind of clunky, a denominator for your sample size. But really, I like to point out the link here. We do have a write-up to kind of explain a few different examples of, kind of slowly increasing the complexity of subsampling and how you would, examples you might use the reporting basis here in like a soil sample assessment, for example, and where you might use the collection basis because you're looking at a certain number of fruit on several different branches of that one tree that consists of your one plot. So just a good thing to kind of work through if you do have more complicated sampling example. It can be a little confusing what goes where, but hopefully that can help kind of clarify, because that especially can be real important to either specifically describe what you're doing, or if you're a protocol writer, it'd be very important to describe what needs to be done. And then that all kind of funnels into the number of subsamples, which is really just how many values are you going to record per plot for that assessment. So in that example where we were going to have a look at 10 different plants and do something with them, then you'll see that changing that to 10 suddenly gives us 10 cells, if I can click in the right spot, 10 entry cells for that one plot to put in the data for. I'll keep it simple here and just do the one. Then the rest of these, just kind of give a blanket statement for, because a lot of this is documenting what is, what's present at the time of application. So we have the stage information, diameter, height, density for both the crop, and then repeat that very same list for the pest as well. So whatever you may need to that's pertinent to document when this assessment is being taken. Then there's a few, you can put in the name of who took the assessment, information about like the equipment. There's some timing fields, so these will automatically calculate based on your rating date and whether it's your, the last application that was documented in this trial or the planting date. It'll just automatically calculate from there. And then action codes we'll utilize later in the data review and see a little bit more about that. So that's really the kind of the description here of what we're doing, with a very, very brief example. Of course, there's 1.5 million different different ways to fill this in, but we'll keep it simple in this case and kind of move on to putting in data into ARMs specifically. And there's not I guess a whole lot fairly obvious, I suppose, much like typing in any program that will just put in a number, press the enter key, and it'll move you down to the next plot where you put in another number and press enter, move to the next cell. What might matter is, of course, the order of the plots. Right now, it's sorted just in plot order, but perhaps that's not the way the data was recorded. So there is a sort up here in the toolbar. So you can sort by replicate number, block number, column number, any of these information. You could sort the table down below here by either ascending or descending. But probably more useful specifically for this situation is to utilize an assessment order or maybe the harvest order. So those are defined on our trial map. I'm going to use this little shortcut button over here to bring up our trial map. And on the map, there's a movement arrows tab at the bottom to define what is our assessment order. So there's a great list of different ways to move throughout the trial. So serpentine within blocks, I will go start at plot 101 and work my way down that replicate. But when I get to 105, I'm not going to walk all the way back. I'm just going to hop to the next plot, which is 205, and work my way back and serpentine through. And then there's try to cover any combination if it's the exact opposite of that. Maybe I'm going across. So there's serpentine across blocks instead. All sorts of different options here. I'll just pick, I think we started with across blocks. So start at 101 and work our way up for whatever reason. Harvest order is the same set of options. It just allows you to choose 2 different ways to move through your field. Most commonly, a person on foot taking an assessment, and then maybe the combine at harvest time, but it could be used for whatever. And when you set that order, then you can sort by assessment order, either on the toolbar or this shortcut button right here will also do the same thing. You can see that changed the order of our plots to match that order on the map. So now this might be easier. I'll delete my other assessments, and this is going to more closely match the piece of paper where I wrote down the assessments on. And so now I can cruise and fill in my values. I wanted to highlight here, I entered a 33, which is outside that minimum or maximum range. Just wanted to point out kind of the purpose of that range is so that way if you type in something that's not valid, ARM will stop you real quick and prevent you from getting too far down the road and having that wrong value. So, okay, you have to be between 0 and 30, but you filled in 33. So, okay, oops, that was my mistake. I just needed the 3. I can say, okay. Or if actually it is 33, you're not stuck here. You can always cancel, and that gives you a chance to go back and change that. And maybe in our case, we just totally need to eliminate our range because, of course, it makes no sense that we'd have a maximum number of plants that could be in the field or what have you. Some quick keyboard shortcuts, I guess, while we're here. There's the single quote key. So that for me is right next to my enter. I can't even really show it on the screen. I guess I could use it in my description. Just a single quote is all I'm typing in. And that will copy the value that's right above that cell that I'm currently in. So I'll press that again, and I only press one key, and it has copied 33 from up above, and it also pressed enter for me. So you can see I can go real quick if you had a column of all zeros or a stretch of all 100s or whatever, real quick way to fill in the same value over and over again. There's shortcuts for 0 if you type the letter Z or the letter O. That'll give you a 0, and it's also pressing enter for me right away. 100, C for centi, or H for 100, or I think also, because it looks like a 1. So those are all kind of just little shortcut buttons that you can press to fill in values. One of the nice things with ARM is an assessment point here. We'll just take this last 100, for example. Doesn't have to be limited to just a number. Over on your properties panel at the right, there's a section devoted to the assessment where there's more that you can add about this particular rating. So there is just a text field for a comment. You can put in, extra details about, something about that assessment. If something seemed kind of off, a little strange, or if they're, whatever you might have that you want to jot down, and this will be attached specifically to this one value right here. There's also room for a barcode. You can record GPS coordinates specifically for that value. You can mark it as damaged, which will toss it out of the analysis, basically treat it like a missing data point. And you can also attach an image. So if you're taking plot photos, this isn't a very realistic picture, but we'll just take it. You can attach a photo, and this will put it right on that plot 105 specifically for that column. You could actually take 20, 30, 50 photos all in a row, and this wizard here will kind of help you to match up the order that you took them. And ARM will even rename those image files for you if you'd like, and can include your trial ID, treatment number, assessment date, plot, things like that. And you have full control over how that's named. That can be a big time saver to bring into ARM, associate it specifically with the assessment value that you have, and then kind of catalog them as well, renaming them to something kind of useful and identifying. So that's all they're linked to specifically that one cell. And you'll see I put in a comment, so there's a little red triangle. That's kind of the symbol here to see that there's something else more here, kind of like in Excel. It's kind of the same thing. And in this case, it was that comment that we added. All right. Well, I think that mostly covers just the real basics of getting some data into ARM. Kind of the next step then, once you have the data in, is to, I guess the term I use is data confirmation. Trying to separate it from data review as two different entities. Firstly, once you've filled in numbers from your piece of paper, or if you're taking it right in the field and putting it down into the software, we want to make sure that that transcription was correct. And so there's some tools to really just confirm the data that we're putting in here to make sure that it is accurate, correct. We didn't have typos or anything strange here that happen as we're putting it in. I'm going to close this trial just because we've got a bunch of junk here, and it'd be a little more useful to review an assessment that's been taken already. So I'm just going to switch over to this trial that actually has an assessment kind of filled in. So we'll pretend I just got done, typing this all into ARM and wanting to look at, okay, how can I kind of look over this data that I just put in? And so there's two really good tools to kind of visualize and look at these numbers here. And you'll find both of them on, again, this assessment properties panel at the right. And it's the section of tools. I'm going to actually work my way up. The bottom one here of these three is the assessment map. And this will draw the trial map, but instead of coloring by treatment or coloring by replicate, it will create a heat map of the assessment. So it'll take your smallest assessment value, it'll take the largest assessment value, split it up into about 10 different groups at maximum, and make a heat map where light colored plots have small assessment values and dark plots have large assessment value. And so here, I guess there's a few different uses, I guess, for this map here. The first one is just to see extreme values one way or the other. And in this case, probably pretty quickly, your eye probably jumped to 904. Pretty obvious something a little weird is going on with him. He is so much darker than any of the others. And if you click on a plot, it'll actually color by current treatment, so you can even see how treatment 4 performed across the other replicates. And they were all very, very modest, except for this replicate 9. So something doesn't seem right with that value. So what we want to do then is go and look at 904. So if I close this and maybe sort by plot number, so it's easy to find. All right, 904. And sure enough, here I have a 120 that's been entered. Whereas, looking at our assessment, it's a percent area, so it's actually physically not possible to have 120% of our leaf area filled with disease. So that can't possibly be true. So that's kind of one of the goals here in the data confirmation is to catch a mistake like this and catch an outlier that really this is an outlier. This value in our case is not physically possible, but in a less extreme case might just be not real feasible, very unusual. And so then of course we have to decide kind of what to do. And really it's going to depend on the situation here. 120 we know can't possibly ever be true. So maybe I can go and look at my sheet of paper and see, oh yeah, it's just 20. I just typed it in wrong. Or what if my paper is smudged? I don't really know what it was because maybe it's a 12, maybe it's a 10. You can't really play guessing games. So if we can't just confirm exactly what it should be, that's where that damaged marker can come in handy. So marking that as damaged, if I do that here on the properties panel, we'll put a red line through the value. And so what this will do is really treat this plot as a missing data point for this column. But we still can maintain what we recorded, both the value and any comment we have. And it would be wise to put in a comment to, at the very least, explain why it was marked as damaged. Maybe we cannot confirm real value, but 120 is not possible, or whatever explanation for that decision. It would be wise to kind of include in that. But just because it's unusual, of course, doesn't mean you can't use it. So obviously, the decision has to be made depending on your situation, what to do with that value. But here in our case, we'll just throw it out, say there's nothing we can do. This data was taken a week ago, and my only record is smudged or long gone, or whatever I did with it. And so we really just have to toss that out and live with that kind of performance hit, if you will, when we get to the analysis. So I'm going to return to the assessment map here. And actually you can see now that with throwing that value out, we actually get a little more interesting map now. That value was so extreme, it was really washing out any of the other differences in all of the plots. And so now things are less extreme. We still kind of have one plot that's really jumping out at us. But just looking at some of the options here, this is the same control as the trial map. So if you're familiar with the show true scale, we can turn that on here as well. So instead of just little squares to represent each plot, we can actually draw to scale what the trial would look like using the plot width and length and some of the layout settings for buffer zone and things like that to really get a good spatial look at what the trial would look like. So this can be handy to kind of spot side effects and things like that maybe wouldn't jump out immediately just looking at the list of numbers. So a really nice tool, again, kind of tucked away a little bit here on the properties panel. This was the assessment map. All right, the next, the other good tool here for kind of confirming the data and just kind of taking a preliminary look at it is the box whisker. So this will draw a box whisker graph of the assessment. If you're not familiar with that graph, just some real quick definitions. Here we have each different treatment. So we're graphing per treatment in this assessment. And we have the 75th and 25th percentile of the data makes your box. And then you have the anything kind of outside that range becomes a whiskers out there. You can kind of see these little red dots. They're pretty small, but if you can spot them, that's actually the data points themselves. So you can kind of see how they end up making these whiskers. That's our largest value and the smallest value down here. The solid line here is the median, where that would be your 50th percentile, right smack dab in the middle of the distribution. And then we can also display the dashed line, which is your mean, your average. So some things to kind of utilize or to really visualize here. You can compare the heights of the boxes, so that the height of each box kind of gives you a rough look at the variance. in each treatment. So just to kind of compare and notice that, wow, treatment 3 was a lot more consistent, a lot shorter box than the rest of these. And we'll care about that a little more in the data review when we start talking about the assumptions of ANOVA. So we won't get too bogged down with that, but just to give a good sense of what the variance is. You can also look at the skewness by comparing the median to the mean. So in this case, actually both of these cases, you have the solid line, the median is lower than the mean. So that shows that the mean is getting kind of pulled up by a larger value. In this case, we can see there's a 40 way up here. That's kind of pulling everything up. Same thing over here with treatment 3. There's a 25, it looks like. And that actually is an outlier because there's an X instead of the whisker. So the outlier takes the height of the box. I think it's 1.5 times the height of the box. If that's that far away, then it's considered an outlier, at least by the calculations of a box whisker. Whether it's something we should throw away or not, of course, we should look at the data and decide that. But at least gives us an idea. And as I hover over it, I can see it's plot 705. So that would be another one to maybe spend a minute to go look at what that value is and decide if it's correct or not. And if it is, maybe document more what happened in this plot. Why is it unusual relative to the rest of them? So again, a nice visual to just get a preliminary look at the data kind of as it's getting put in to make sure we're not overlooking something obvious while it's getting put in. All right. So that was kind of a fast look at the data confirmation as well. Any questions through all the stuff we're kind of firing rapid fire a little bit? All right. So the last subject I wanted to cover, we covered our data entry. We just did our data confirmation, kind of 2 tools there. The last one, really thinking logically through our little storyline here. We've filled in our data. We made sure we filled it in well without any typos or strange outliers. And now maybe we have in 3 more days, we need to perform this same assessment. What's the next action that we would do for that? And I think most often, the thing most people are familiar with is, oh, I have to do the same thing again just a few days later. Well, I'll just copy this column and I'll paste it, and then I'll update it with whatever's different. Well, instead of doing that, we can formalize and standardize that process by using standard evaluations. So these SE files, I think it's a terrible name. It's really hard to train on this because it feels super complicated. But I think if we just had a snazzy name for it, maybe it would sound easier. So, but bear with me here. An SE file is really taking that description of an assessment. This is what we did. In our case, when we were filling it in, it was a stand count. Really basic example, but it's a stand count, and we do those all the time in our trial work here at whatever company I'm at. Instead of filling that in every time by hand, it took us 15-20 minutes with me talking through it all to fill it in. Instead of doing that over and over again, why don't we standardize that and automate that? And that's really what an SE is offering. So in SE, you get to choose 1 or more columns that we've defined. You get to specifically choose which rows from that header. So you don't have to take them all, but you can choose which ones are always going to be the same. And we get a bonus too if you're utilizing study rules. We have a bonus that'll actually just come with as well if you're using those in any of your columns. Didn't really touch on those, but just kind of a nice bonus for us. And so, reasons to learn SEs, like I said, it sounds hard. So it's hard to get people excited to try them out. But if you're a protocol writer, if you're planning out experiments, then you really want to standardize your data across all the trialists, across your company. And so SEs are by far the best way to do that. It's really taking I want to fill in all of my column headers in a protocol, so that way all of my trialists do it the same, and taking that to another level so that all of the protocols that I write, all of the protocols my whole company writes, have the same description, which leads into the goal of a multi-trial analysis. If everything that's done the same is described the same, it can be pulled together very easily, and you can get to summarizing and combining that data and looking across all those trials run very quickly, instead of dealing with the mess of data being described and in different places everywhere. If you're a trialist and say, well, yeah, that's not a big deal, I'm just trying to get my trial done. It is a form of automation that will save you time. It's not something now you have to fill in each and every time. You do it once. You've described this thing once. Now let's stamp a name on it. It's in a file. I just have to click load and grab that file, and there it is. So I think there is a time saving as well. And then it's going to make your trial more valuable because it's more consistent. And especially if that SE came from your protocol writer, if it came from your sponsor, now you're describing your data the same the way they want it to be done. And now suddenly that trial that you're handing over is so much more valuable to them, because there isn't any more time they have to spend mucking around and getting it to look the same as everyone else's. It's ready to go, so it can all be combined, and that makes them extremely happy. So I guess just to real quickly kind of walk through then the creating an SE. Like we said, our example was, all right, we just took this rating. I guess it's not my stand count anymore. This is the other trial, but we filled in and performed this assessment. Now we want to do it again. It's easy in this one trial to copy and paste. I probably can't convince anyone to say, well, an SE is so much better than copy and pasting in this one trial. But if I'm going to do the same assessment in my next trial, now do I need to open 2 different ARMs, and I got to copy and paste to make sure I get my windows right, so I'm not copying from one into the wrong one, et cetera, et cetera. I think you can start to see where you can save some time. So let me delete this column. Say this was the assessment we cared about. So if I want to package this column up, to be able to use again and again, I'm going to right click and export an SE. So the first question is, which columns are part of this file? So I'm just going to choose this first one, but one nice advantage to these SEs is they could have multiple different columns. So if there's 4 assessments that you do all together at one time, that you repeat again and again, that really has a lot of advantage now to roll that all into a single file that can then be used again and again. But I'm going to just do one for simplicity's sake. Then the next question is, what rows from this description do we want to save? So I'm going to clear this list just to start with. Text box down here, you can type in the number that corresponds to the row, or you can just click and choose. Okay, well, do I want to include this pest? Well, probably not, because I could do this assessment for any type of weed that I'm going to encounter, right? In this case, it's a disease trial. So it could be any disease that we're dealing with. I really don't want to hard code that disease in, but maybe all I work on is winter wheat. That's the only thing I'm ever have an experiment on. So let's include all of those. It can really be customized to your own needs, but for sure I'm going to want these three. Maybe my sample size is important in this case, maybe not. You're really just thinking what rows, what of this information is always the same every time I do it. And if it can be different, then maybe leave it out of the SE and you can fill it in each time you load. So there can be a little bit of strategy to it. Definitely wouldn't want the stage majority, maybe the stage scale. If I'm always using that crop, then that might matter, but I'll leave that open there. So you get to choose what components matter that you want to repeat. And then it will save it onto your computer. And you can give it a name. So you want to be kind of descriptive of what you're bundling up here. I guess, plant count, or this was, percent area. So maybe I'm just going to call it percent area. Whatever is that you are going to recognize. The name itself doesn't matter in the grand scheme of things, other than it's what you're going to see in the list when you're choosing what to load. So I'm going to be very, very boring, and percent area of disease is what this is called. And then the file description, that could kind of match up with whatever you'd want to describe it to be. You have some more characters here if you wanted percent area, disease, whatever you want to fill in to describe it. But this will create, essentially, it's kind of like a virtual clipboard, if you will, that we've copied only specific rows of this column, but we've copied it, and then we'll be able to paste later in whatever trial we're in. From here on out, doesn't matter. I'm going to have that clipboard, if you will, copied. It's just saved to a file on my computer that I can load again and again. So maybe actually I'll close this trial, because like we said, a good example would be that we're not in the very same trial. I'll go back to the first one we were in. I want to perform that same assessment in this trial. I can right click and I can insert that SE that I just created from the file I created. And this is the list of all the different SEs that I've created on my computer. Just a little file here. And if I open that up, just the rows that we chose. So I notice I don't have any hard-coded pests or crops, because this study might have an entirely different one that we're dealing with, but it has just the components that I chose. We basically pasted into this study. And I can repeat that again and again if that's what we're doing today, and I have to do it in another 3 days. I can just, once again, insert and choose to load that. So it's kind of a template for your assessments. There's a lot more you can do with this, but I kind of want to keep it simple. I know we're about running out of time, but just real quickly, if you don't quite know where to start. So, today, the very first example, we just started from scratch to fill in an assessment. These SEs actually give you an opening to a common question I hear of, well, how do I know what to fill in? There's an alternative to just starting to fill in what you're doing. If you go to the SE definitions, it's a section within your site description. These do tie in with the SEs you've loaded, but I am just going to insert a new one in a trial that you haven't dealt with SEs before. This will be empty. You'll just have one blank one there to utilize. And if you go into the SE name, there's actually a list here. And there's a favorites list of SEs you've used. Of course, if you've never done this before, this will be empty, and it'll take you to the full list of SEs where we have, I think it's 700 or 800 SEs. One of the large global sponsors shared their list of SEs. And they kind of code the SE name. I think the letter is kind of the discipline. So those will be insects. That's probably like a fungicide. But I think the real benefit is just looking at that description. So if you just want to know what should I do for a stand count? Well, come in here and you could filter on the description for stand. And so here are a list of different SEs that at the very worst could give you an idea. On the right, there's a preview. You can see how this, plant stand looks like they're basing it per hectare. So they were using a reporting basis of per hectare. This one's maybe more plain, just plant stand and how they use to describe it. You can either just get inspiration of what to fill in, or you could actually utilize this. Maybe this is exactly what I want to have. This has a plant stand, and then there's a calculation even in a separate column to standardize it per acre. Whatever you want to do, you can actually use these. If you say, okay, I'll actually kind of bookmark that one as one I want to use in this trial. And then when I'm in the assessment data, if I wanted to use that one that I bookmarked, here in assessment 3, perhaps, I just click into SE name and it'll bring up that list. And this is one that I bookmarked for this trial. Just like we saw before, it loads in all of this information that was saved in that file, so I can do it the same every time. Just kind of another example here. All right, I've got a question, and we are running kind of full on time here. So I'll try to get to this. And if you have any other questions, I can stick around, but I don't want to hold anyone hostage. The question is to navigate around the SE list. The up and down arrows toggle the view up and down. It doesn't move the cursor up and down. So we said that is the SE list in the assessment data. So is that when we're in the SE name here? The up and down arrows toggle the view up and down, but it doesn't move the cursor. Yeah, it looks like even if I'm moving, I'm trying to press the keyboard up and down. I see what you mean. It's not moving the selection. It's just moving the screen. I don't have enough of them, so I'd have to be really short for it to do what it's asking. But I see what you mean. Instead of just being able to press the down arrow or something to pick this second one, I have to move over with the mouse. Got you on that one. Great comment. That's definitely something I can pass along to the programmers. I can't imagine there's a special reason it behaves that way. I guess if there is, then I'll learn something tomorrow. But otherwise, yeah, that would make a lot of sense. I'll just make a note of that and kind of dig that up, because yeah, that is suboptimal. Good catch there. All right, well, we've kind of hit our hour mark. I appreciate all of you joining us today. Like I said, I can stick around if there is a question, but definitely don't want to hold people hostage. Yeah, Melissa?
No, not me, Matt.
I'm sorry, what was that?
No, I didn't have a question.
Okay.
But if other people do, yeah, jump in now. Now's the time.
I appreciate the attendance today. Hopefully we'll get to see you all, each well, I guess Friday for you guys. It's late Thursday here, so I gotta do that mental math. But to cover these different topics. And yeah, so appreciate the attendance. And if you think of questions during the week, then you kind of hold on to them then, and I'll try to talk a little less next time around, so we have a little more time at the end too, if you have questions. So,
or if you have a question that comes to you during the week, feel free to send it through. And if I can't answer it, then I can send it on to Matt or the support team in the US.
All right. Very good. Well, thanks everybody. Then, without seeing questions, I guess we'll close up shop.
Thanks, Matt.
All right, thanks.